Okay, well, thank you. And I'm happy to introduce Dr. Jungwei Lee, who is an assistant professor at USC's Keck School of Medicine. Jungwei uses innovative approaches towards kidney regeneration and disease modeling using stem cells. And the title of his talk is Novel Stem Cell Tools to Model Polycystic Kidney Disease. So welcome, Jungwei.
Thank you, Bino. So I would like to thank Dr. Terry Watnick for giving me this wonderful opportunity to share my lab's some of the research tools. So as a stem cell biologist by training, my lab is interested in development of novel stem cell tools for the kidney research community, specifically for researchers to study kidney development and disease, and eventually towards the rebuilding of a synthetic kidney for transplantation. So if we want to make best use of stem cells, we need to first get to know how the kidney normally develops and then make use of the information to guide the in vitro efforts. So specifically in the mammalian kidney organogenesis process, there are two types of cell populations that are guiding through the whole organogenesis process. One cell population labeled in pink here is MM, metallithic mesenchyme, and another cell population is called UB, urethrial bud. So at the initiation of kidney development, the UB would invade into the surrounding MM and start the branching morphogenesis, eventually generating a collecting duct, while the MM receiving signals from the UB will be induced to form nephrons and stroma. So with further analysis of the MM and UB, right now we know that at least 4 different progenitor cells are within UB and MM to drive this process. These include nephron progenitor cells, shown in purple here, the NPCs, that generate nephron structures. And in close adjacent to the NPCs, at the budding tips of the UB structures are the ureteric progenitor cells, UPCs, expressed in WNT11, which self-renew and generate the whole collecting duct system. And at the most outer layer of the MM, close to the NPCs, are one layer of interstitial progenitor cells, IPCs, that generate stroma populations. In addition, there are vascular progenitor cells that generate the vasculature of our kidney. Because of the central roles the different kidney progenitor cells plays, my lab is centered on the studies of kidney progenitor cells, developing new tools for the progenitor cell culture and expansion, and eventually use them to study kidney development and disease. In particular, right now, we have been able to capture the NPCs in vitro in culture and generate nephron organoids to study nephron development and disease. We can also capture the UPCs in vitro and generate collecting duct organoids to study collecting duct development and disease. At the same time, we are combining NPCs and UPCs together to reconstitute the normal normal kidney organogenesis process towards the rebuilding of a synthetic kidney for future transplantation. So I will first introduce you our NPC system, nephron organoids, and the use of them for PKD modeling. So 10 years ago, when I started my postdoc training at the Salk Institute with Dr. Juan Carlos Ispixua Belmonte, in the kidney regeneration field, there's no way to generate any of the kidney progenitor cell types. So with the stem cell training, I reasoned that because NPCs are constantly self-renewing during kidney organogenesis process, we should be able to identify a culture system that can recapitulate the niche signals and maintain the NPCs in vitro so that we can get an NPC9 expandable. So towards that goal, I screened different growth factors and small molecules, eventually identified a chemically defined medium that can support 3D expansion of NPCs. So those NPCs can be expanded stably for several months without losing their potential. In vitro, those cultured NPCs can generate nephron organoids, and ex vivo, when the cultured M-cherry labeled NPCs were mixed with the wild type UB, you can see that the M-cherry labeled cells would cluster just right around the body UB tips to reform the nephrogenic niche. If we inject the M-cherry labeled cultured NPCs into the developing kidney, you can see they would form chimeric kidneys in vivo with the normal wild type kidney cells. After I started my own lab at USC, we further improved the MPC culture system. Right now, we have a advanced MPSR V2 medium, which allows not only the culturing and establishing MPC9 from purified 6-2 positive MPCs, but also it can generate MPC9s from isolated MM or even from the dissociated whole kidney cells of early embryonic kidneys. So those MPCs can stably be expanded and they can generate all segments of the nephron in the form of nephron organoids. So in the human system, we have used CRISPR-Cas9 to engineer human stem cells with a 6-2 GFP and PAC2M cherry dual reporter. With this dual reporter system, we were able to purify from direct differentiation from human stem cells, the MPC population. And then we have a unique human MPSR V2 system medium to generate and derive human MPC9s. Again, with the robustness of this culture medium, right now, even from mixed cell types with only 30 to 50% differentiated from the human stem cells, we are able to derive a stable MPC9s that can be expanded for a very long time. After more than 100 days, you won't lose their developmental potential. Importantly, after culture, the transcriptome appears to be more closer to the primary MPCs compared to MPCs derived directly from the human stem cells. And importantly, those MPCs can form highly patterned nephron organoids with the formation of glomerulus and the various types of renal tubules formed in an organized way. So inspired by Dr. Bino Friedman's pioneering work in human stem cell-derived PKD organoid system, we wondered if we can also use our MPC-derived nephron organoid to mimic PKD. Towards that goal, we used a one-step, highly efficient multiplexed CRISPR-Cas9 knockout system to generate PKD2 mutants. So we selected 2 of the individual clones that has double or neo PKD2 knockout for further studies. So because those MPCs can be long-term expandable, we first developed a scalable system to generate thousands of nephron organoids. So we first seeded those MPCs as spheres using agri-well system, in which we can produce thousands of spheres in one single well of a 6-well plate. So with those thousands of spheres, we seed them into a shaking culture system, and we observe that when the MPCs start to differentiate into nephron organoids, after 3 to 4 days, we start to see the emerging of cyst formation, and then those cysts grow bigger and bigger in the next few days. We characterize those cystic organoids in terms of PKD features and found that actually those cystic lining cells are expressing much higher phosphorylated histone H3 signal, suggesting that they have more proliferative cells present. And consistently, cell cycle relevant genes, mTOR signaling genes, and MYC activity were dramatically elevated in those cystic organoids. And even the metabolic reprogramming described in PKD in vivo is partially recapitulated in the organoid system. As you can see here, compared to the control organoids, PKD2 knockout cystic organoids appears to be using much higher OCAR and ECAR to use the maximal energy consumption to support their rapid proliferation. As you can see, as a control, without differentiation, PKD2 knockout in the MPC stage, they won't have such a big difference in metabolism. So we also carried out a proof concept drug screening using our scalable PKD modeling system, and we chose a small library consisting of hundreds of small molecules targeting specifically the epigenetic pathway. And here, as you can see, the screening is very, very robust with consistent results from 2 individual MPC9 bearing PKD2 knockouts. Importantly, as you can see, the identified hits belongs either the HDAC inhibitor group or BRD4 inhibitor groups, both of which have been described before as PKD treatment in mouse models. Interestingly, we identified one novel small molecule called PTC209, which belongs to the BMI1 inhibitor, which BMI1 is one component of the PRC1 complex of the epigenetic machinery. So interestingly, this analogs of this drug has been used in clinical trials for cancer considering the similarity between PKD cyst lining cells and cancer, we envision that this small molecule or BMI1 inhibitors could have some potential effects in treatment of PKD. So in the human model system, again, we can introduce PKD2 mutants and generate those expandable human NPC9s. And using a similar shaking culture system, we can get these scalable models of PKD in the human. And in human, specifically around day 8, instead of day 3 or day 4 in the mouse, we can see big cyst formation, which grow bigger and bigger. So these organoids grow, so 100% of the organoids would form cysts. And the protocol is very simple. We just aggregate them and then using 2 different medium to drive the differentiation, and the cyst would form in 100% of the organoids. Importantly, I noticed that those organoids can be frozen and thawed, so that allows us to deliver the organoid resources to researchers who have no access to organoid techniques, so that we can maximizing the potential of using organoids in the whole PKD research community. So on the other hand, my lab is interested in developing similar systems that can capture the collecting duct development. So starting from the mouse models, we have manually isolated E11.5 UB from the mouse embryo, and we have identified a cocktail of medium that can expand the UB tip cells in the form of UB organoids. So from each individual UB tip, we are able to generate hundreds of homogeneous UB organoids, which allowed us to identify a differentiation condition that can generate those highly elongated collecting duct organoids with the presence of both aquaporin-2 positive principal cells and the FOXI1 and the ATP6V1B1 double positive integrated cells in the same tubule across the entire collecting duct organoids. In the human system, we also developed a very robust protocol in which from adding human stem cell line, we can generate these KIT positive UB precursor cells, and then using a specific UB culture medium, we can fully program them to UB organoids, which is highly expandable. After 6 months of expansion, they still keep homogeneous Wnt11 GFP, suggesting these UB organoids are almost near pure UB tip cells. So we have proof of concept to show that genome editing can be directly done in the UB organoids to mimic rat knockout congenital kidney disease phenotypes. So more recently, we have started with UB organoids to further develop a connecting duct organoid model. And as you can see, following the current protocol, we can get a beautiful elongated human collecting duct organoid from the UB organoids. Importantly, by profiling this system across organoid differentiation process, we found that it clearly transitions through the normal UB differentiation process. Like at the beginning, it is showing highly enriched expression of the UB tip genes like Wnt11, RET, GFRA1, and the ETV group of transcription factors. And then after around 7 days, it gradually transitioned to the UB trunk expressing elevated ELF5, EHF transcription factors, and IRF1 also transcription factor, as well as the Wnt9b, which is a Wnt ligand driving the nephrogenesis. And then we found that at the end point, they would turn on all kinds of principal cells and integrated cell gene expression. We confirmed this gene expression profiling by performing whole mount immunostaining. As you can see, the integrated cells are clearly sporadically distributed along the aquaporin-2 positive collecting duct tubules. And importantly, we identified the homogeneous of expression of transcription factors such as H1F1B along the collecting duct epithelial cells with the presence of aquaporin-2 and ENaC subunit exclusively on the apical side, which is a lumen side of the organoids. So Dr. Staraschenko's lab from University of South Florida has used patch clamp assay to record the activities of potassium channels, sodium channels, and chloride channels, all present and active in the intact connecting duct organoid, suggesting the mature and functional features of our connecting duct. And more recently, we used our PKD2 knockout human stem cells, as described earlier, to generate the UB organoid and the CD organoid. And again, we found that the cysts can form in our mature and functional human collecting duct organoid. Currently, we are trying to characterize these collecting duct models of PKD extensively using the assays we described earlier for the nephron organoid model of PKD. At the same time, we are trying to generate an even more complicated kidney organoid model. We call it a synthetic kidney, by mixing MPCs and UPCs together so that we can generate both nephrons and collecting duct in the same structure. So as a proof of concept, we have mixed our expandable MPCs and UPCs together. As you can see here, UPCs are labeled green. If we mix the single cells of MPCs and UPCs together, you can see after 3 days, MPCs would cluster to the center and start to invade and branch out into the surrounding MPCs. And after 3 weeks, we see dramatic branch morphogenesis. And by immunostaining, we confirm that we can form around 150 nephrons in each of the synthetic kidneys. And the interesting thing is that we can frequently see, even from the bright field view, the deposition of phenol red in the synthetic kidney tubules after around 2 weeks of synthetic kidney formation, suggesting the maturation of the synthetic kidney. Surprisingly, in addition to the formation of nephrons and the collecting duct, we found the formation of variety types of stroma cells, including interstitial cells, mesangial cells, smooth muscles wrapping around just the central collecting duct network, and the renal capsule cells expressing NIV2 exclusively on the periphery of the synthetic kidney structure. So this was surprising because we didn't include IPCs, the cell type, the progenitor cell types that normally generate stroma. So interestingly, after a lot of analysis of lineage tracing, we noticed that the cultured NPCs became plastic by losing PAX2, critical transcription factor. And this subset of NPCs can spontaneously transdifferentiate to FOXD1 positive IPCs and migrate to the periphery of the structure. And eventually the formation of the complete nephrogenic niche containing 3 different progenitor cells generate a whole collecting duct system, nephrons, and stroma. Dr. McMahon helped perform single-cell multi-ohm sequencing to confirm the presence of nephrons, collecting duct, and the variety types of stroma cells in the synthetic kidney. And by comparing the different stages of embryonic kidneys and the postnatal kidneys and adult kidneys, eventually we position the synthetic kidneys in between postnatal day 0 and the postnatal day 9 kidneys, suggesting these kidneys already transitioned to a postnatal stage, which is relatively mature, even not as mature as an adult kidney. So we confirmed that several of the highly important transporters or functional molecules in the synthetic kidney are expressed at similar levels to a mature 3-month-old adult kidney, and even the subcellular localization of critical transporters like LRP2, SLC12A1 are exactly the same as that observed in adult kidney sections. And we also performed a large number of functional assays, and one of them shown here is for proximal tubule functionality, which is blocked by a specific transporter inhibitor.
2 minutes.
We also identified the different endocrine functions, for example, the secretion of urine with the presence, the activation of cyclic AMP, and the producing of vitamin D3 metabolisms in the presence of vitamin D3 substrate together with parathyroid hormone. And if you put the synthetic kidney into hypoxia chamber, they would produce erythropoietin. We transplanted the synthetic kidney in vivo and found that it formed big cysts filled with fluid. This fluid is actually highly enriched in urea and creatinine, suggesting the production of primitive urine. And we also confirmed by whole mount staining, showing the formation of nephrons, vascularized nephrons. And we also found the cyst lining cells are exclusively expressing GATA3, suggesting the collecting duct origin of the cells, indicating the urine produced from the nephron are eventually connected and stored in the collecting ducts with enlarged collecting ducts showing cyst formation. So this is our summarized model of our synthetic kidney. So eventually, what we want to investigate further is that can we model PKD using those more advanced synthetic kidneys that have functional nephrons, collecting duct, and the stroma to investigate PKD pathogenesis. So with that, I would like to thank my lab members, my collaborators, and my funding agency. Thank you very much. Hope it's going to be useful for the PKD research community.
Thank you. I'm not sure if we have time. Maybe we have a couple questions, maybe a minute or two for questions.
I think you have 5 minutes for questions.
Okay. So a couple questions in the chat. One is, do you think that 3D printing of kidneys is possible using your approach?
Yeah, that's excellent question. Actually, we are doing that. So recently, I was very lucky to get a NIH Director's Award to support us to make a prototype synthetic kidney towards transplantation. So currently, I'm working with Dr. Xiaochen Chen from UC San Diego, who is a bioengineer specialized in 3D printing and biomaterials. We are trying to use the 3D printing techniques to scale up production of the synthetic kidney, as well as to make an exit route for the urine to drain, and so that we can test this prototype for improved kidney function in vivo.
Okay, question from Dr. Delling. Did you also generate PKD1 knockout cells, and do they form cysts in your system?
Yeah, excellent question. So we have generated PKD1 knockout in the mouse NPC9s, and they form cysts. And we tried to generate PKD1 knockout in the human, but because the antibody is very difficult to detect using Western blot to show if PKD1 is knocked out or not. So currently, we don't have the resource to test PKD1 in our nephron organoid models of the human. But we will maybe working with Bino to try this system.
I had a question about forskolin. Are you using forskolin in any of these assays?
Forskolin, no, they don't need forskolin. They would spontaneous for cysts in the shaking culture setting.
And if you add in forskolin, does it make a difference?
We tried that. It didn't increase forskolin. It didn't increase larger cyst formation.
Okay, there's a question. Fantastic talk. Were you able to assess whether there's a decline in synthetic organoid function after cyst development? For example, with EPO or urine production?
Oh, okay. So we didn't transplant the synthetic organoid cyst. Oh, we didn't transplant the PKD knockout synthetic kidney dish in vivo to model PKD yet. But that's of interest, and I think we should try. Thank you for the suggestion.
And there's a question about what the polarity of the cysts is.
Okay, so we haven't done that yet, but I feel it could be very likely to be the same as Bino's model, because I feel if there's no matrix, I feel maybe the primary will still facing the outside, facing the median part. Yeah, in the normal organoids we produced, we have tried the primary cilium. It's always facing the lumen side. It's regular. Yeah, and because our connecting duct organoid is cultured embedded into a matrigel, so most likely those organoids would have the normal polarity of the cyst.
What is the source of the vasculature in the grafts? Is it organoid or host?
Yeah, so we have known in our model and also in other kidney organoid transplantation models that the vasculature would form from the recipients. It's from the animals.
Can you or have you titered cells with correct PKD into the PKD knockout organoids to understand how much function can be recovered?
Oh, not yet. We haven't done that yet.
And did you try whether your culturing system with shaking makes a metanephric kidney cyst?
You mean like ex vivo kidney? So I guess the question is if we try the ex vivo isolated embryonic kidney, if they form cysts. I think we didn't try that experiment. We did try to put the ex vivo culture into the transwell and add phosphocholine, and we reproduced what people observed cyst formation in that setting.
All right, well, there's lots of questions, but I think we've got to move on. So thank you very much.
Thank you, everyone.
All right. So I will go ahead and introduce our next speaker as she prepares for her screens. So next we have Dr. Kristen Bernard. She is the Elizabeth Mears and House Jamieson Professor of Psychiatry and Professor of Genetics at Yale University School of Medicine. Her current research combines expertise in human stem cell models, genomic engineering, and neuroscience to identify the mechanisms that underlie brain development, traits, and disease. The title of her talk is Using Stem Cells to Explore the Genetics Underlying Brain Disease. Take it away.
Hi, thank you. I'm going to have us change gears here for a little bit and stop talking about the kidney. Not because I don't think the kidney is a fantastic organ, but because I think a lot of the tools that all of us are using in our different organ contexts can really inform the questions and the tools that we all bring to our own studies. And so with that, I kind of want to just talk about what we're doing in the brain, and I'm going to hope that you see some similarities to the open questions that you are all studying in the kidney, and maybe ways for all of us to collaborate down the road. And so to start with, I want to start by saying that psychiatric disorders are common, severe, heritable, and genetically complex. And so I think, you know, with that in mind, they're not so different than, you know, the same reasons that you are studying the kidney. For those of you who don't think about the brain every day, I want to really explain what I mean by heritable and genetically complex. And so schizophrenia is perhaps the most heritable of the psychiatric disorders, at least based on twin studies, where estimates of heritability are about 80%. Now that heritability is not fully explained. Genome-wide association studies that are looking at the common variant risk structure underlying schizophrenia can explain about a third of that heritability. Rare variant studies are adding another maybe 5%. And so much of this heritability is still unknown. And when we consider it across the spectrum of psychiatric disorders, you see that we have schizophrenia, autism, attention deficit, hyperactivity disorder, and bipolar at the top, all with over 75% heritability. And at the other end, we have disorders like depression and post-traumatic stress disorder that are more like a third heritable. But from the top to the bottom, again, we have, you know, about a third of the heritability being explained by common variants, and much of it still not being explained. And so by diving in more to schizophrenia, just again to explain all of the types of genetic risk that has now been associated with schizophrenia. The green dots are copy number variants. So these are rare deletions found in less than 1% of cases, highly penetrant, but typically pleiotropic. So implicated not just in schizophrenia, but also autism, developmental delays. And so these are on the rare end of the spectrum, but highly penetrant. Likewise, more recently, genome-wide whole genome sequencing has identified almost a dozen protein truncating variants. Again, even more rare, about 10-fold more rare, but just as penetrant, linked to schizophrenia. But in aggregate, all of these rare variants, again, explaining perhaps less than 5% of the known heritability, and most of the genetic risk for schizophrenia being in these common variants of small effect. Now, each of these common variants imparts less than a 1% increased risk for schizophrenia. In truth, all of us were born with a dozen or more common variants for schizophrenia. Patients were just born with a few dozen more, on average about 60 common variants for schizophrenia. When we consider common variant genetics, we end up with these Manhattan plots, like I'm showing you on the right. For those of you who are not familiar with Manhattan plots, each vertical line is really composed of dozens or hundreds of single nucleotide polymorphisms that are all inherited together in a cluster at that given region. Everything over that red genome-wide significant line at 10 to the -8 is a loci significantly associated with disease. For schizophrenia now, we've got about 250 of these loci. That being said, despite the hundreds of thousands of people who have been, genotyped, it is extremely hard to match each of these vertical peaks, these loci, with the gene or genes that they're regulating. Again, the basic hypothesis being that they are potentially in enhancers or 3' UTRs, regulating expression of nearby or more distal genes. We also don't know how these variants interact in some, and again, they're not inherited in isolation. All of us inherit a dozen or more of them. And finally, we don't know how in aggregate they impact clinical penetrance and expressivity of the common variants or even of the rare variants that are inherited with them. It's been really challenging to study psychiatric disorders in mice. For one thing, their brains are, structurally different. For a second, their behaviors are fundamentally different. What does it mean to have psychosis as a rodent or social deficits? But finally, at their fundamentalness as a model, rodent models can be challenging to model non-coding common variants because they're infrequently conserved between humans and rodents. They can be challenging to dissect the complex interactions between variants because it's difficult to engineer and breed mice with dozens of variants together. And finally, it can be challenging, again, to look at things like pleiotropy and variable penetrance and expressivity when mice are all on the same background. And so, and these are, again, exactly the types of variants I think that are most relevant to psychiatric disorder, non-coding common variants inherited in combination with like clinically meaningful but difficult to describe differences across individuals. And so we've really been asking in the lab, how do we relate genetic variants to their impacts? And this is the classical idea, right? That we have a genotype linked to a disease and we want to understand the phenotype. But the question that I'm actually much more interested in the lab is, can we modify genotype-phenotype relationships, right? So we all know that there are certain environments that can make phenotypes greater. In the context of psychiatric disorders, these could be traumas, things that happen, or in the, for example, in the context of schizophrenia, it seems like a lot of cannabis use can make clinical presentation more severe. In the context of neurodegenerative disease, it's aging can make phenotypes more severe. And it goes without saying that if there are environments that might make phenotypes more severe, there might also be phenotypes that make them less severe. And these are the questions that I'm really interested in. How do we modify the relationship between the DNA that we're born with and the clinical impact? And by environment, I'm talking very broad here. We're talking about the rest of the DNA around that variant. We're talking about the cell type that the variant's acting in, and then we're also talking about context. What hormones, what chemicals are being exposed to the cell. There's a long-standing track record of looking at environmental influence in psychiatric disorders. One example is this maternal immune activation hypothesis, which really goes towards saying that infection or stress in the mother in second trimester increases the risk of psychiatric disorders 20 years later. And we see this in times of war, in famine, and we can see it in rodent models as well. Secondly, we also understand that there is a time period in adolescence where individuals are particularly prone to second hits of stress and drug abuse that again might exacerbate the phenotypes they would otherwise have. Our first foray into looking at these gene-environment interactions was led by Rachel Yehuda at the Bronx VA in collaboration with the New York Stem Cell Foundation. This was a really ambitious study that Rachel encouraged us to take on, where we generated stem cells from 39 individuals. All of them were combat-exposed veterans at the VA. Those stem cells were turned into glutamatergic neurons, and then we stressed those hormones by treating them with HCORT, the human stress hormone, hydrocortisol. So here I'm showing you some basic RNA-seq performed by Karina Sau, where we're looking at high and low dose response to stress. And as you can see, the more you stress the neurons, the more their gene expression changes. The types of genes that change expression when you stress neurons related to neurodevelopment and immune response, all this makes sense. But what I didn't tell you is these 39 donors that were combat exposed, half of them had PTSD and half of them did not. And so you can subset that same data, now break it down by diagnosis. And you can ask how do we distinguish neurons that come from PTSD cases or controls? And in fact, we distinguish them not at high dose stress, but at low dose stress. All neurons respond to high dose stress, but the PTSD derived neurons were hyper responsive to stress, and they responded more robustly at low dose, to the extent that we could distinguish neurons from cases and controls at low dose H-court. And the types of genes that were responding at low stress in the cases, but not at low stress in the controls, are already linked to PTSD through their postmortem RNA-seq and through genetic studies. But the genes were not specific to PTSD. We also pulled out genes linked to developmental disorders, schizophrenia, epilepsy, and autism, really showing us that stress is dysregulating broadly across psychiatric risk genes, not just PTSD genes. We can take this further and begin to look at cell type specific stress responses. So what I first showed you is how glutamatergic neurons respond to stress, but we generated an equal data set looking at how GABAergic neurons, inhibitory neurons, respond to stress. And we can cross this with what we know about the genome to ask which specific loci have regulatory activity in stress context, but not at baseline. And then we can ask of those gene targets, what diseases are they enriched for? And so we see big enrichments across neuropsychiatric disorders, but I think more interestingly, we also see them across inflammation and metabolism. Again, saying that, it's important to consider not just how genetic loci act at baseline, but how they act in response to environmental context that might inform your disease of interest. And so, what I think I'm showing you here is that we think H-court is an example of something that might make phenotypes more robust. And I didn't have time to show you, but we think that sex hormones potentially might be an example of an environment that might have the potential to make hormones or genetic risk less robust. And that's, for example, where we look at autism or schizophrenia, where rates of disorder are 5-fold more common in males than in females. And we think there's a potential role for sex hormones in minimizing the penetrance of those variants. Now, I talked to you about how risk variants sum with the environment, but they also sum with each other. And this is something we've been looking at for a very long time, started by a former PhD student, Seokmin Ho. We're looking here at some of these common variant target genes that we upregulate and downregulate. We can look at RNA-seq. We can look at the impact on synaptic function. But more interestingly, we can manipulate them in combination. So here we're taking the top 4 GWAS target genes, manipulating them one at a time for RNA-seq, computationally adding these RNA-seq together to yield an expected additive model, and then comparing that with the actual experiment in the lab when we combinatorially perturb these genes all together. When you do this, you can ask how good your expected additive model is, and the answer is actually pretty good. About genome-wide, about 82% of genes are changing as expected when you manipulate them in combination, but 7% of genes are more down than expected, and those genes that are more down than expected are enriched for all the major neurotransmitter classes. Reciprocally, 11% of genes are more up than expected, and those genes that are more up than expected are enriched for the rare and common variant genes linked to schizophrenia and bipolar. And so what I'm really trying to say here is that we see emergent biology when we manipulate genes in combination that you could never have predicted by manipulating them one at a time. And this is so critical, I think, for all complex genetic disorders where risk actually reflects aggregate impact of many risk variants. And the only real big caveat to this study was that we prioritized these 4 genes on the strength of the genetic evidence. These were the top 4 EQTL genes in the GWAS, but 3 of them happened to have synaptic roles. And so Michael Deans in the lab went back to revisit this across a larger number of genes, specifically asking the question about the role of biological pathways in how genes come together in combination. So the first study was based on Common Mind Consortium brain collections and the PGC2. This follow-up study was based on a larger collection of postmortem brains and a larger GWAS. And so rather than having 4 risk genes come up, we ended up having 25 risk genes to come up to choose from. And so Michael took the top 5 of those 25 genes with a known synaptic role, the top 5 of those genes with a known epigenetic or regulatory role, and then the top 5 genes that were not synaptic, not regulatory, and had no apparent relationship to each other. And then he repeated the design, manipulating these genes alone and in combination and asking how well the additive model predicted the combinatorial effects. Now with that synaptic set, we actually exactly reproduced the previous results. About 20% of genes behave non-additively. Likewise, in the regulatory group, we have about 20% of genes not being predicted by the expected additive model, but the multifunction group was completely additive. So all genes in combination, in general, change as expected by the single gene perturbations. Now, these non-additive genes in light blue, when we tried to figure out what the biology was, what is unique about these genes that are not changing as expected in the additive model, well, they were actually enriched for the convergent genes that were downstream of the individual 5 synaptic genes. And so what I'm saying here is that if you manipulate the 5 synaptic genes one at a time and you look for shared downstream genes changing in the same direction in all 5 individual perturbations and call those the convergent genes overlap to a large extent with the non-additive genes. This is also true with the regulatory genes. Again, a huge number of the convergent genes are being observed in the non-additive group. And this multifunction group that had no non-additive genes also had very little convergent biology. If you want to ask more questions about the convergent genes, we can show that they, just like the non-additive genes, are extremely enriched for the rare and common variants linked to schizophrenia risk. And that they enrich for the biological processes linked to schizophrenia too. So glutamate receptor activity, GABAergic receptor binding, synaptic function, and so on. So again, these are experiments I've shown you thus far are based on arrayed experiments, 1 gene changing per well. But in order to validate this result, we followed up with a pooled design. So here in 1 well, we're manipulating all genes in combination. The goal being to get 1 CRISPR guide RNA into each cell, and then sequence that guide and the single cell RNA-seq profile for each cell and put that back together. And so using this approach, we were able to validate the results across 17 schizophrenia risk genes, some of which came from the original subset of 15. Here's an example of the convergent network we saw in the synaptic list and the convergent network we saw in the epigenetic list. And I show you this because we saw strong convergence in each, but none of the convergent genes were overlapping between either set of genes. Following up again, here's the full convergent network from the arrayed experiment, again enriched for schizophrenia risk genes, and likewise the convergent network from the pooled experiments. To summarize what we're showing here, if you think about the individual gene perturbations for manipulating the top 10 genes linked to disease, for example, you might see different effects with each gene. And when you put them together, you create the expected additive model. And what we're observing experimentally is that the measured combination is actually a little bit less than what would be expected by the additive model. Likewise, when you look at these individual perturbations and ask what genes are changing downstream of them in a common direction across all the individual perturbations, we find these convergent targets, and those convergent targets are explaining some of this non-additive effect. We wanted as a final act to ask if convergence, downstream gene expression changes in the same direction, could predict drug responsiveness. There's a bit of a precedent for this in psychiatry. So here is work done by Matt State's lab where he screened for drugs that could reverse a single autism gene, DYRK1A, and then showed that it was the estrogen receptor family that could reverse the DYRK1A phenotype. But not just DYRK1A, but 2 other autism genes, Norexin-1 and ADNP, were also rescued using estrogen receptor agonists. Again, coming back to this idea that sex effects modulate outcomes in autism and other psychiatric disorders. Likewise, Lorenz Studer's lab was able to show that Wnt signaling could rescue some, but not all autism risk gene effects. And so Kayla in the lab, co-mentored by Laura Huckins, likewise tried to computationally predict drugs that might reverse our convergent signatures. The first thing I'll point out is that she pulled out the stress hormone, glucocorticoid again, that I talked about earlier in the context of PTSD, as hitting these convergent targets. We pulled out one drug, atoximir, that we then tried to use to reverse the effects of one of our genes of interest, CALN-N1. This is a lot of data here, but the important thing to look at is the original RNA-seq where we have 2 tails, a tail up on the left and down on the right. These are our RNA-seq effects from the single gene perturbation. And when we add the drug, we really lose these tails. We're normalizing expression across the profile. And you can see the same thing again here with atoximir and TMM-219. So this is far from saying that we've cured schizophrenia, of course, but it is suggesting that we might be able to predict convergent targets that themselves might not be schizophrenia risk genes, but are downstream of one or more risk genes. And so the idea here, overall, big picture, is that we hope that gene-gene and gene-environment interactions will inform genetic resilience, helping each of us achieve our best genetic potential. And for the grad students in the audience, I want to quickly highlight this really exciting program that we're running at Yale, where we're looking to identify, recruit, and retain exceptional and diverse recent PhD graduates for structured promotion to faculty at Yale. And so the stipends are huge. The mentorship will be fantastic. We want to bring you to Yale now after your PhD and see you succeed here as a faculty member 5 years later. Please don't hesitate to reach out if you have questions. And with that, I want to highlight the members of my lab who've done the work, the funding sources that sustained it, and to thank all of our many collaborators. So yeah, I'm happy to take any questions as time permits. Thank you.
Thank you. Let's see. So I'll check the chat. Or if anyone has questions, they just raise their hands and we can get to you. I'll start off. I had one question about, you mentioned that the patients with PTSD were hypersensitive, and that was shown in comparison to patients without. And so my question is, when were you able to scale lock if patients were already diagnosed, were they treated with medications, and do those medications contribute to the sensitivity, or are they in opposite kind of effectors?
This is such a fantastic question, and I think the real answer is that it's complicated. There's been a long history of linking stress response to PTSD. It's not clear clinically whether patients are hyper responding to stress or failing to turn down the stress signal. And we're definitely, modeling a very small part of a much larger thing. We also don't really know in truth whether we're totally modeling an adult exposure or just even a fetal developmental exposure. I think it's hard to know whether neurons respond the same way developmentally to stress. So we're really just asking, how do neurons respond to stress? We're way underpowered in this cohort to match it to clinical outcomes. We're now going back and trying to double the size of the cohort, which might get us closer to these answers. I do hope that it might mean, like we could imagine having, ambulances maintained with, fast acting drugs to, change your stress response. Like this could get to a way of, helping prevent PTSD following trauma, but I think it's a very long way from being there yet.
In the chat, we have, what percentage of these convergent genes were themselves in loci linked to psychiatric disease?
Yeah, that's a great question. I don't know the percentage off the top of my head. I do know they were enriched for risk loci, so that was some of the plots that I showed. I also know independently, I've worked with Doug Rudifer and Mike Talkowski, who have looked specifically at convergence in autism, and again, actually predicted novel autism genes that were subsequently identified to be autism genes. And so, yes, some of these convergent risk genes are just underpowered autism risk genes that will come up. And some of them represent maybe new biology or genes that we can't tolerate full mutations on and still yield humans, right? Because we do have that selective pressure that some of these genes that are important in the brain might be more important in the heart or the kidney, and thus, require developmentally in other cell types. And so not showing up as autism risk genes.
And I'm not seeing anyone else in the chat, but I'll ask one more. In comparison, when you showed versus an adolescent-induced stress, so like stress that has been kind of inherited over from birth to age, versus whether a child in stress or constant stress, do you have any idea of the difference between like a child maturing under constant stress versus an adult firstly experiencing stress and then that being kind of carried out?
This is such a good question, and it's so important to life, right? Because stress and trauma doesn't happen randomly. There are people who, are going, for a lot of reasons are disadvantaged and will have, stress in both childhood and in adulthood. I don't know I don't think any of us truly knows what the difference is biologically and mechanistically between childhood and adult stress. I do know I've heard some, really intriguing findings. For example, if you look at an adult cohort and try to predict who's going to have PTSD in it, the best predictor of adult PTSD following an adult trauma is actually which amongst them had a childhood trauma as well. And so there is definitely something sensitizing about childhood trauma, but humans being terrible test subjects, it's also important to keep in mind that, one of the greatest things that happen to children as traumas are parental neglect or abuse. And that tends to be something that reflects how they were raised too. And so you have this genetic risk and environmental risk that tracks with kids over generations. And so it can be really, really challenging to disentangle genetic and environmental risk when they're so entwined.
Yeah, and I think that was excellent. Also, the fact that in animal studies, a lot of times you see stress or incidents are conditioned, and so actually they are less sensitized to it. So with adults, it's a difference in prolonged trauma to see you're more desensitized versus spotty trauma as being more sensitized. So that was excellent. And I have one more question in the chat. In pooled CRISPR screens, how do you obtain a precise quantitative representation of gRNAs?
Yeah, so I mean, this is just sequencing based, right? So we're just literally sequencing the guides in each cell. We're going with an MOI of about 0.5. So the hope is that every cell either has 1 or 0 guide RNAs. And so we're using eSite-seq, but other groups are using Perturb-seq and Crop-seq, with the idea being that we use the guide itself as a barcode, but other groups can have separate barcodes and guides. But you sequence the guide, you sequence a single cell, and it's all in the same droplet, so you can put it back together computationally.
Thank you.
We can move on to the next speaker.
Now we're going to move on to the abstract presentations. These are 5 minutes and 5 minutes for questions. And the first speaker is Dr. Shohei Kuraoka, who's an MD PhD postdoc fellow at MGH and Harvard Medical School.
Thank you for introduction. This is the can I share my slide?
Yeah, that looks good.
Okay. I'm honored to speak in this symposium. Today I would like to talk about my new candidate pathway of the ALPK disease genesis. So, as you know, the ALPKD is a seriopathy characterized by the formation of severe renal cyst and the progressive decline in kidney function. The causative gene for most ALPKD cases is polycystic kidney and hepatic disease 1, PKHD1. Cyst formation derived from the distal nephron is characteristic in ALPKD. However, previous mouse and organoid models have shown cysts derived from the LTL positive proximal tubers. And so improved experimental models for ALPKD need to be established. In a recent study, we developed an organoid-on-chip model with PKHD1 mutants. In this model, the organoids exhibit the cystic phenotype in LTL negative but CDH1 positive distal nephrons under flow conditions. Based on transcriptomic analysis in this organoid-on-chip model, we found that LAC1 is highly upregulated in patient renal cysts. And 3 LAC1 inhibitors have shown therapeutic effects in the human organoids. To understand the mechanism by which LAC1 influences formation in ALPKD, we analyzed the RNA-seq data derived from human kidney organoids carrying PKHD1 heterozygous and homozygous mutations. LAC1 activation and deactivation are regulated by low guanine nucleotide exchange factors, GAF, and GTPase activating proteins, GAP, respectively. In our data, GAF were decreased, and there was no notable decrease in GAPs. Oh, sorry. These data led us to consider another mechanism, such as reduced consumption of active LAC1. Then we evaluated the expression of 27 LAC1 effectors that were previously reported. Interestingly, the majority of these effectors were downregulated, which is consistent with the accumulation of active LAC1 in ALPKD cysts. In-depth exploration of these 27 effectors
2 minutes.
Combined with data from existing literature and human kidney databases, revealed only one effector, SP-TAN1, SP-TAN1 potentially associated with cystic biological processes and distal networks. SP-TAN1 causes alpha-2 spectrin, and this is a filamental cytoskeletal protein known as an essential scaffold protein. Immunostaining of kidney organoids mirror the expression pattern of SP-TAN1 in the human kidney, demonstrating its predominant localization in distal nephrons. Quantitative image analysis revealed a significant reduction of this protein in distal nephron in the organoid model. The reduction of SP-TAN1 was also observed in cystic epithelia of ALPKD patient kidneys. To validate this finding, we investigated SP-TAN1 transgenic mice. Surprisingly, the CDH1 positive distal nephrons show dilation. This is similar to ALPKD patient and the organoid model. To further investigate the role of LAC1 SP-TAN1, we generated transgenic organoids of hetero and homozygous epistemal knockout. We hope to elucidate the mechanism of ALPKD cisgenesis mediated by this pathway for future therapeutic development. In summary, the organoid on-chip model and fluid flow culture recaptured the cystic phenotype in distal nephrons in ALPKD organoid model. LAC1 activation in ALPKD is driven by the reduced expression of its effector of SP-TAN1, contributing to the development of pathological cystic processes. Ongoing investigation include the confirmation of this phenotype using SP-TAN1 knockout pluripotent stem cells and further analysis of mouse transgenic models. These findings may offer novel insight into pathogenesis of ALPKD, potentially leading to novel therapeutic avenue for ALPKD patients.
Time.
This is the last slide. Thank you.
Okay. Thank you very much. Questions? We have 5 minutes. I can start with one. I thought that was interesting. So, you know, the RAC, what is known about SP-TAN1 as an effector of RAC1? You know, is it an inhibitor of RAC1? Is that the model?
Thank you for question. This is a very difficult question. The SP-TAN1 was reported as the effector of LAC1, but my detailed function is it is not clear yet.
There is a question in the chat.
Oh, I see. Okay. Why do you think the non-tubular flow is facilitating cyst formation in the chip model?
Yeah, that is a very good question. So in our chip system, of course, there is the outside mechanical stimulation, but even in the inside of lumen was some stimulation was existing. So of course, this setting was different from the Vivo setting, but our system can mimic the mechanical stimulation for the kidney organoids. So that stimulation can cause the cyst formation.
Yeah, it's interesting that the heterozygous mouse has a phenotype.
Yeah.
You know, do you have any insight into what would happen in a homozygote, or, you know, how that heterozygous phenotype is actually working?
Yeah. So the first, when we noticed the SP-TAM1, so I searched the SP-TAM1 mutant mice database, but so there is no published about the SP-TAM1 transgenic mice. So and one mouse database showed the only heterozygous mutation of SP-TAM1. So maybe I guess the homozygous mutation of SP-TAM1 caused the embryonic lethal. So, but I have to check the further investigation about the transgenic phenotype.
Dr. Germino asks, do the heterozygous mice have a liver phenotype?
2 minutes.
Yeah, thank you. The good question. So honestly, so I don't know. So because the mouse database have a very small information. So still, we could investigate only kidney tissue. So we have to check the other phenotype of other organs. Yeah, thank you.
And maybe this is the last question. Inhibition or activation of RAC1 is reported to have an effect on CFTR. So did you check in your system if CFTR was changed?
Yeah, thank you. So we have not, but yeah, we will check thank you for suggestion.
Okay, maybe time for one more question if anyone wants to throw one out there. Okay, otherwise, let's thank the speaker and move on. Thank you. Okay, I'll introduce then the last speaker out of order, and that would be Matthew Cavanaugh from the University of Kansas Medical Center. And he is going to talk about O-gluconacylation as a modulator of metabolism and ciliogenesis, promotes ADPKD progression.
All right, is my sharing working? All good?
Yes.
Good.
Excellent. All right, well, thank you very much for the introduction, and thank you to the organizers for giving me the opportunity to share what I think is some pretty interesting data. So before we talk about O-gluconac, we need to consider how it is produced. O-gluconac is a post-translational modification whose precursor molecule, UDP-gluconac, this rainbow here at the bottom, is produced via the hexosamine biosynthetic pathway. It is then attached to its substrate proteins via O-gluconac transferase and removed by O-gluconacase. Now, what I want to bring your attention towards are three particular metabolites: glucose, glutamine, and acetyl-CoA. And note that all three of these are elevated in P basis for the metabolic changes of PKD, prompting elevations in O-gluconac. Now, it's been shown in other disease systems that persistent elevations in these precursor molecules can produce O-gluconac at abnormally high levels, and this in turn can have adverse effects on cells and whole tissues. Additionally, it's been shown that in both in vivo and in vitro systems, either genetic or pharmacologic inhibition of O-gluconac transferase's activity can lead to shortening of the primary cilia, the topic I'm sure some of us are familiar with, and its potential role in PKD pathogenesis. So previously, our lab has shown that in both juvenile and adult onset models for ADPKD, there is elevation of O-gluconac, particularly in the cis lining cells. This was confirmed on immunohistochemistry and whole kidney lysates with Western blotting. Now, what I've been doing recently is the generation of a new juvenile onset mouse. This is a conditional knockout under the control of the HOXV7 cree, which is going to be deleting PKD1 and or OGT from the collecting ducts, starting at about embryonic day 10.5. Now, by postnatal day 14, we can see by the histology here, there is a significant increase in cystogenesis in our PKD1 knockout, but that is significantly less severe in our PKD1 OGT double knockout. And these beneficial changes are reflected in reductions in cystic index found below, kidney weight to body weight ratios on the right, as well as blood urea nitrogen as a marker for kidney function in the lower right. Additionally, we've gone on to perform some survival studies with this same mouse line. The reason that we considered P14 as our time point was, as you can see, this sharp decline in mouse survival between weeks about 2 and 3
2 minutes.
meant that we couldn't wait too long. And with the exception of one double knockout mouse that haunts me in my dreams, we've seen an amazing improvement in survival as a result of this intervention. We found partial resolution of numerous misregulated signaling pathways based on our whole kidney lysates, particularly the phosphorylation of CREB and ATF1, the expression of CREB, the phosphorylation of AMP kinase, as well as the expression and phosphorylation of STAT3. Additionally, various markers for proliferation, the nuclear marker PCNA. We have alpha smooth muscle actin for fibrosis, particularly myofibroblast activity, and F480 for inflammation via infiltrating macrophages. All of those are significantly elevated in our PKD1 knockout, but in our double knockout, we can see across the board beneficial reduction in all of those. And this corresponds to a resolution of the abnormally elongated cilia that we also see in this model being reeled back in to a more normal level. And finally, just so you know that it isn't exclusively a mouse phenomenon, I wanted to show you some patient histology here on the left, showing elevated ogluconac in the nuclei of cyst lining cells here marked with DBA. And when we did some 3D in vitro assays to generate cysts using patient-derived cells, administration of OSMI1, this is a drug that inhibits OGT, was able to get us a decrease in both the number and size of those cysts. I think that's all I've got. So I'd like to thank everyone involved, our lab, our collaborators, and the various organizations and foundations that helped fund this. And with that, I'll take any questions.
Okay, perfect timing. Questions? I have a general question. One might imagine that interfering with ogluconac or knocking it out could affect many types of physiological functions in an animal. So, are the mice okay in general? If you forget about PKD, do they have any phenotype on their own?
Yes, so I'm going to go back a few slides just to our survival. Now, we have not dissected any of these mice, although we have passed over half a year with no overt changes to their, exterior appearance or their behavior. But I will be interested to see as we reach the end of this type of study, will there be any significant changes? Our P14 time point indicated very little in the way of difference. I mean, you can see minor elevations in PCNA not reaching the level of statistical significance, but we do see slightly shorter cilia in our OGT knockout as well. But on the whole, they seem to be doing well, which is a very good sign, I'd like to think.
And there's a question in the chat. Do you see changes in body weight gain over the time course of the study suggestive of systemic effects?
I have not been monitoring weight over time, which, well, now I'm really kicking myself for having not done that. Now, particularly in our juvenile mice, as they're getting up to postnatal day 14, we've been tracking their weight and have found that the PKD1s tend to be underweight. The OGTs don't seem to be significantly different in weight, and the double knockouts, similarly, no major change. It's just those PKD1s that come in underweight, which further exacerbates that kidney weight to body weight ratio.
And then there's a question, have you looked at the OGT knockout in whole animals or with a more ubiquitous CRE versus the HOXB7 CRE?
Now, a ubiquitous knockout of OGT is incompatible with life. I believe it's embryonic lethal, which does throw a wrench into the idea of therapeutics, unless we can get one of those more targeted administration routes. But yes, it's not going to work if you block everything.
I have a question. Looking at the OGT deletion and you have your increase in PCNA, did you also look at an increase in cell cycle arrest, seeing that OGT leads to kind of that transactivation leading to cell cycle arrest? Do you know if that's upregulated?
We have not taken a look at that yet,
2
though I
minutes.
think that would be a good companion experiment to these PCNAs. So, well, when this gets published, be on the lookout for it, because that might be in there.
There's a question whether you can inject the OSMI1 into animals.
So OSMI1, this particular drug is not fit for injection as is. It is not sufficiently soluble. However, we do have a potential collaborator. Oh goodness, I wish I remembered his name right now, but he's been working on a similar drug that can inhibit OGT, but is soluble in PBS. And we'd be interested to see where injections could go for that.
Have you tried any other Cre-recombinase models such as PAX8 to see whether you would have a different effect in a slower onset model?
Read my mind. We actually do have a PAX8 mouse that we're studying right now, induced from 4 weeks of age out to 6, doxycycline in the drinking water. But as you can see, I've already crammed a bit too much data into this 5-minute talk, so didn't get to that, but we will have some adult data coming up in the coming months.
And last question, were the double knockouts born at the expected ratios?
Approximately. There was a time where, you get a couple litters and you don't feel like you've gotten as many as you ought to, but I did track them over time and it came out roughly Mendelian. And I should mention that OGT is an X-linked gene. Probably should have mentioned that earlier. Hence why you could get different distributions between males and females.
Okay, great. Thank you.
You're welcome.
Okay, I'm so sorry for the inconvenience caused. Good afternoon. It's my great honor for me to present my study in RRC. Today, I will talk about my project about targeting ALDH1A1 together with nanoparticle-mediated kidney-specific immunotherapy deleted growth in ADPKD. So firstly, I will introduce some background. ALDH1A1 is regarded as a cancer-stem biomarker in cancer treatment. It can promote the cancer progression, therapy resistance, and immune evasion. ALDH1A1 can be regulated by epigenetic factors include BRD4, EZH2, and mRNA. Our group has reported BRD4 can regulate cis growth in ADPKD. We are wondering whether the ALDH1A1 plays a role in ADPKD. Therefore, firstly, we investigated the expression of ALDH1A1 in PKD1 mutant cells and kidneys examined by Western blot and RT-qPCR. Importantly, we found that inhibition of ALDH1A1 with its specific inhibitor DSF delays growth in PKD1 mutant mouse kidneys as seen by decreased cyst index, kidney weight-body weight ratio, and immune levels. This treatment with DSF can also delay cyst growth in early stage of the ADPKD mouse model. Importantly, the treatment also decreases cyst lining epithelial cell proliferation as examined by Ki67 staining. In addition, treatment with DSF deactivated PKD associated signaling pathways such as DAS3, S6, and AKT in PKD1 mutant epithelial cells and kidneys. So we further found that treatment with DSF can decrease the recruitment of macrophages and the level of CD4 positive T cells, but increase the level of CD8 positive T cells as examined by FO18 staining and flow cytometry. So Recently, immunotherapy is very popular in treatment with different diseases with PDO, anti-PD-L1 or PD-1 antibody. However, treatments with anti-PD-L1 antibody alone do not efficiently disease growth in ADPKD models. We are wondering whether the co-treatment with other drugs can increase the efficiency of PD-L1 mediated immunotherapy. Answer is yes. We found that co-treatment with DSF and PD-L1 antibody significantly disease growth. So to understand the mechanism of why treatment with ALDH1A1 inhibitor could synergize PD-L1 antibody-mediated immunotherapy on disease growth, we found that ALDH1A1 can serve as a transcription factor to directly
2 minutes.
regulate PD-L1 transcription as examined by Westbroad, qPCR, and ChIP-AC. Our results suggest that immunotherapy combined with chemotherapy could be more efficient on disease growth in ADPKD patients. So in the past years, the nanotechnology has been significantly developed, and the nanoparticles has been used as a drug carrier to test in different models of diseases. Ms. Paul's silica nanoparticles are promising and no drug carriers. So our next question is whether using nanoparticle carrier to deliver PD-L1 antibody can disease growth. To increase the specific nanoparticle in kidney, we conduct kidney-specific categorizing antibody together with anti-PD-L1 antibody. MSN, our results are very encouragement. We found that treatment with nanoparticle conjunct with PD-L1 and CDH16 antibody disease growth in PPD1 mutant mouse kidneys as seen by decrease of kidney weight body weight ratio below level. We further confirm that CDH16 conjunct with nanoparticle only enter kidney and livers, but not other organs, as examined by in vivo imaging. In summary, the upregulation of ALDH1A1 play an important role in ADPKD progression. Targeting ALDH1A1 either alone or together with PD-L1 antibodies disease growth in PPD1 mutant mouse models. Treatment with nanoparticle mediated PD-L1 and CDH16 antibodies disease growth. Our study suggests that PD-L1 mediated immunotherapy combines with chemotherapy and nanotherapy should be more efficient in ADPKD treatment. In the last, I would like to thank my mentor, Dr. Lee, and my lab members. I would also thank the collaborators and the funding support. Thank you. I would like taking any comments and questions.
Okay. Checking the chat. I will start one question from the chat. We have, what type of kidney cells did the nanoparticles target?
We use our nanoparticle conjuncted with the category 16. We can be targeting the renal epithelial cells.
We can't hear you. Can you get closer to the microphone, please? I'm not sure what happened.
I believe it was renal epithelial cells.
Yeah.
Yeah, I was able to hear that. And then my second question is, what did you guys look at like a PKD2 knockout? Is it the same?
PKD2 knockout, we haven't tried, but thank you for your good suggestion. We plan to try PKD2 knockout cells of animals. It's a good suggestion. Thank you.
And your next question is, how were the nanoparticles delivered, and what portion or proportion of the kidney cells were successfully targeted?
Nanoparticle delivery. We do the IP injection, and in some few studies, we will test the category 16 levels through the IHC staining or F staining after injection the nanoparticle. We also did the in vivo imaging to see the accumulation of nanoparticle in different organs.
I think there was one question before that one.
Yes, I see it. I missed that one. Does your ALDH1A1 inhibitor increase acetaldehyde levels in your mice?
Oh, thank you for the good question. We plan to detect this this levels in future studies.
And then you have how long were those mice treated with the nanoparticle? And is silica biodegradable?
Oh, yes, that's a good question. We treat the mice for 2 months, and the silica is is about
2 minutes.
biodegradable, because we found that when we take a different in vivo imaging to see the accumulation of nanoparticle, we can see the change of nanoparticle in the liver and the kidney.
Okay, perfect. We are at that 1 minute mark. If there are any more questions, we have time for one more question and we will end this. Okay, that'll be it for our part. Thank you so much, Alice.
Thank you.
So I just want to thank everybody today for your participation. Thank you to all of the speakers. We had great sessions. Thank you for to the moderators. So we are going to adjourn for today, and tomorrow, the second day of the symposium, will start at 9:50 in the morning with a few more exciting sessions for tomorrow. So thank you to everybody, and we'll see you tomorrow, hopefully.